Now we welcome Professor Yaakov Nahmias of the Faculty of Science at the Hebrew University of Jerusalem, Dr. Symon, the head of Radiology at the Sheba Medical Center, and Yaky Yanay, President and CEO of Pluristem, who will talk about their innovative treatment for the virus. They will be interviewed by Maayan Hoffman, news editor and senior health analyst at the Jerusalem Post.
Well, hundreds of research teams around the globe are vying to create a working vaccine. Many others are working to save lives through innovative treatment protocols and drugs. Today, we have with us some of these leaders, people who believe that they may have found a method to reduce the severity of COVID-19, to keep people off ventilators and out of the hospital. Welcome, Professor Yaakov Nahmias, Professor Symon, and the CEO and President of Pluristem Therapeutics, Yaky Yanay.
Hi.
Let's get started. Researchers at Israel's Hebrew University of Jerusalem and New York's Mount Sinai Medical Center believe they could potentially downgrade COVID-19 severity into nothing more than the common cold. Professor Nahmias, you are one of these researchers. You recently revealed that the FDA-approved drug Tricor could reduce SARS-CoV-2's ability to reproduce or even make it disappear. Explain how you came to this conclusion. How does it work?
So one of the interesting things about viruses is that they're actually parasites. They can't replicate on their own. It's a small box of protein with a single strand of genetic material. So to replicate, the virus has to go into the human lungs, get into the cell, and then change it in a way that the cell itself, our own cells, start producing new viruses. And one of the things that we studied is exactly how the virus is changing those lung cells. And it turns out that the virus causes this massive upregulation of fat, so massive accumulation of fat in patients' lungs. And that fat is critical for the virus life cycle. So the virus needs it to make new membranes, to make new particles, and then infect the rest of the body. And it turns out that the way it's happening is by shutting down the cell's ability to burn down this fat. And we found that a molecule called fenofibrate, known under the trade name Tricor, is actually very, very effective at reversing that trend. Essentially allowing the lung cells to burn down this fat in the lab, in a Petri dish. It's very important to point that out. And then you don't see this massive damage to the lungs, and you don't see this production of viruses, and the virus production gets shut down. So this is very important, and we actually posted it online, but the next step has to be a clinical study. So from a point of view saying this is a new mechanism, we never saw this before, everybody around the world, let's look at it, and then let's try to get some clinical data to figure out what is happening, and then put together a well-formulated clinical study to see if this can be effective. There's some sort of a road between here and there. Of course,
and we're going to talk more about those clinical studies coming up, but I want to move to you, Yaky, for just a minute. Now, when you learned about the COVID-19 virus, you immediately thought to yourself, how could we use our already PLX cells, our existing treatment, in order to try to benefit patients who are suffering from COVID-19? The PLX cells have already worked in other indications, and now you're seeing that they are actually helping to reduce the severity of the disease. Tell me about these PLX cells and what you're doing.
Great. So PLX cells, these are young, potent placenta cells that we are able to collect after a full healthy delivery. The mother actually is donating the placenta, and we developed in the last decade the process to duplicate the cells in a very efficient way. We're using the product that actually looks like that. It's very simple, cells in a vial. We're using it, and we treated hundreds of patients in the last decade, from cardiovascular to muscle damages, radiation damages, and we have a very good and a long experience treating human beings with these cells to create the body own regeneration. And when COVID started, it was around February or March, I got a call from my friend in China. And he told me, Yaky, we were using the cells which are similar to what we use to treat the patients in order to control the overactivation of the immune system, ARDS. This is the main reason that kills human beings due to COVID. And we said, when we introduced the cells to the patient, we were able to show a very good and quick balancing of the immune system. And this is what we started to do. We started in March. We applied to the Ministry of Health in Israel. We started to treat patients on compassionate use, both in Israel and in the US. Since we had a very good safety profile, they actually approved it very quickly. And in parallel, we applied to clinical studies. And as of today, we are 4 months after the initiation. We have a phase 2 study that is running in the US as well as in Europe, and we are treating patients.
Amazing. And if I recall, that you're seeing a very good success rate, and that is 75-80%. Where are you holding with that?
Exactly. Before the initiation of the study, we did a series of compassionate use. We treated dozens of patients here in Israel and as well as in the US. We were able to show that the cells are reducing the immune response, getting this patient quickly from mechanical ventilation, and the discharge rate was very high, and the survival was very good.
Which is excellent, because the survival rate for people who go on ventilators is not very high. It tends to be 85% who die, if I remember correctly.
That was terrible. The numbers in the US and even in Israel were extremely high, and we were able to reduce and to control that.
Amazing. Now, Professor Symon, you are doing something that is completely new. You are using radiation to reduce the severity of COVID-19. What do you do? Just zap it out of them? How does this work?
Okay, so just carrying on from how Yaky mentioned that one of the mechanisms of death in this disease is there is kind of a huge exaggerated immune response, and this is sending millions of inflammatory cells into the lung, and these cells are actually drowning the patient. And it's called, in medical language, we call this a cytokine storm. And radiation is a very non-specific physical way of breaking the DNA of cells. And the cells that are most hurt by the radiation are those that are rapidly replicating. So these lymphocytes and histiocytes that flood the lung are extremely sensitive to very, very low doses of radiation therapy.
Similar to cancer treatment?
Well, no, actually maybe 1/100th of a dose that one would use for curing cancer. So this would be equivalent to about, say, 10 chest x-rays. And it has been shown, and there are some early studies from some universities in the States, like Emory. There's a study going on now in Mass General Hospital in Boston, Harvard, using a low-dose radiation when the pneumonia begins to get more and more severe and patients are not responding to the other therapies, and then they deliver a low dose of radiation to both lungs, and there have been early reports of some very good responses to this treatment if delivered on time, before it's too late.
Before it's too late, which we tend to hear with a lot of the treatments, I feel like, lately. Yeah. Now, Professor Nahmias, I want to go back to you for a moment. Last time that we spoke, you were actually preparing for some animal trials in New York. Where are you holding with that? Have you kicked off those trials? And what's next in terms of clinical trials that you started to touch on before?
Right, so one of the problems that we have with small animal models, specifically hamsters and mice, is that their metabolism is very different from humans. Mm-hmm. And it turns out that fibrates, very specifically, act completely differently in rodents and in humans. So while animal studies in New York are probably not going to happen, simply because they're the wrong model for this drug, we have been able to get retrospective data from clinical centers across Israel. And the retrospective data looks very, very promising. It looks like patients that are taking bezafibrate, in this case, are less likely to end up in ICUs across Israel. The statistics hold up, but the numbers are still very, very low. So we're still trying to essentially get more retrospective data from several other clinical centers. And luckily, we are pushing into a clinical study, and hopefully that will start in the next few months.
Amazing. And what's fascinating about it is that it's an FDA-approved drug, so if it is proved to be effective, in other words, we'd be able to move forward much quicker than if this was some kind of new treatment or like the new vaccines that we're looking at to prevent the disease. Now, I want to go back to you, Yaky, for a moment, because there was something very exciting, I think, that happened with your company just recently, as well as to the country. Israel and the United Arab Emirates announced that we were going to try to work toward normalization of ties. And with that, Pluristem announced an interesting international partnership in none other than Abu Dhabi. Who are you working with, and how could that help fast track a treatment to COVID-19?
indeed exciting period. I mean, with the COVID and now with this peace process, so a lot of excitement in what we are doing. So I think that one thing about science, medicine, health, and education, it has no borders, it has no limits. We know the center from Abu Dhabi for a while. This is one of the leading centers for stem cell research globally, but in the Arab world, which always fascinated me in personal to know what they're doing over there and how we can build these ties together. And when it was announced about 2 weeks ago, we actually already had some prior interaction with the center, and we decided to collaborate. This center in Abu Dhabi, they actually treated thousands of patients that were infected by COVID. Most of them were at earlier stage. The cells, they're using quite similar cells to what we use, but the cells have the ability to get the immune system respond in a better way to allow the antibodies of the cell or the antibodies to attack and actually to kill the virus. And since both of us are doing cell therapy and advanced therapies, and both of us are doing COVID, we decided to join forces together with the goal that to bring. I think it's time that our region will come with drugs, with prosperity, with well-being, and that's the news that's going to come from our region. So we are collaborating. We signed this memorandum of understanding. Soon we would like to utilize it to advance study in COVID-19. And I'm a big believer in economical peace and building this relationship between the countries and between human beings. So fascinating time. I'm very happy with that.
It's great. Could be peace, prosperity, and no more pandemic.
Exactly.
That would be great. So I want to go back to you, Professor Symon, and ask you a little bit more about the research. Now, I understand you're working with Emory University. Tell us a little bit about that partnership, and also how long until the health ministry allows COVID-19 patients access to such a treatment?
Okay, so we've submitted a clinical trial, and this trial is to actually replicate the trial at Emory University. We are collaborating with Dr. Muhammad Khan, who's the principal investigator at Emory on the study. And the Ministry of Health a few hours ago actually informed me that the trial has been approved, and we hope we'll be able to kick off already next week. And that will be, I will do a ward round in the Corona Intensive Care and the Corona Internal Medicine units at Sheba and try to identify people that are not doing well on the best standard therapy that we have at the moment.
How many patients are there right now at Sheba, or around how many tend to be there within a given week?
Right, there are about 50 hospitalized patients
in a given week.
Probably about 10 or so in intensive care and another 40 or so in the internal medicine wards.
Wow.
Right, but many patients that are doing well are actually sent home. So yeah.
Got it. Amazing. Now, we're sitting around the table. All of you are very trustworthy. You're doing good research, we assume, proper clinical trials. But with COVID-19, we've actually seen a phenomenon where much research is being published before there's a chance for it to be peer-reviewed. And a lot of papers are popping up on Medium and some of these other sites, and we don't know if it's trustworthy or not. Professor Nahmias, maybe you can talk a little bit about that, and how does the average person know what to read, what to trust, and what might be good for us?
So I'm not sure these papers are there for the average person. Let's start with there. I think the fact that academia has moved incredibly fast on COVID-19 is a testament to its strength. We need to take a step back and get everything in perspective. The hepatitis C virus was identified in 2000. The first time it was cultured was 4 years later. The first time it was sequenced was a year after that. The first time we had a drug was 5 years after that. It took a decade to develop effective treatments and to study this virus and understand what it's doing. With COVID-19, we identified the virus, SARS-CoV-2 in November. It was cultured in December. It was sequenced in January. Effective therapies, in tissue culture were available since February. And now there are ongoing clinical studies. Remdesivir was approved just recently. So all this advancement is a testament to the speed and efficiency of not only the new tools that we have, but the academic community, sharing data freely. None of our work would have been enabled without that. But that data is there for the people in academia, the people in the clinic to look and evaluate. the only thing we can really trust are double-blinded clinical studies, period, nothing else. So standard of care should not change without a proper double-blinded study that has been approved, that has been regulated, that has been carried out properly. And regretfully, what we're seeing is politics, not science. And it's more of an issue in the United States than in Europe and Israel, but still, it's important to point out. We're seeing issues that are due to a very embittered political rivalry rather than anything to do with science.
Can you elaborate on that just a little bit? Give me an example.
So the big problem was hydroxychloroquine. It was, the first study done in tissue culture is very effective. Actually, to date, when you're trying to run a screen for inhibitors of the virus, hydroxychloroquine is your positive control. It always works. It works very nicely. It's just that it doesn't work in patients. Now, and there are a lot of drugs like that. We don't know whether fenofibrate will be effective in patients yet. And so the fact that it was published is a very good thing. The fact that even the initial retrospective data and then short clinical study published early was great. And the fact that there was another clinical study after that was double-blinded that showed no efficacy is great. The only thing that wasn't great is when somebody says, well, let's all use it, right? And it's not the right way. But the fact that the data is there is I think is a very effective and the right way of moving forward.
So what we're saying is sharing the data about the medical research is very important with the medical community and whoever is interested, but what we should do in the clinic itself should not change until something has been proven effective.
Exactly. And there is nothing can replace a blinded study.
Got it. So, as we're getting towards the end, I really want to talk a little bit about the idea of a vaccine. The novel coronavirus is not going away. I think everybody agrees that it's going to be around for at least another year, if not more, and that we need to work toward a vaccine. But will a vaccine be a solution for the entire world? Yaky, why don't you start with that?
Well, I have a very firm opinion about it. Obviously, vaccine, that might be a good long-term solution. But I think that today the efforts that we see globally are completely imbalanced. It cannot be that 100% of the effort will be toward vaccination. Drugs and therapeutics and advanced therapies should get the focus and the attention in the coming month and year. Vaccination, even if it's going to be successful, it's going to take any number between 18 to 24 months before we're going to start to see a real effective number of patients or number of people that are getting vaccinated. And I'm a big believer that we should be focused on the short term. And of course, this is something that Pluristem is doing. We are doing treatments. The goal is to take patients out of the hospital, out of the ICU units, send them home. If we'll be able to reduce the severity of COVID, that's something that actually the global system will be able to face it. And I'm seeing there is a lot of pressure globally, economical pressure. People want to go to work. People want to have fun. And I think that the only thing that will be reasonable in the next 12 to 18 months is actually taking drug with repurposing and allowing to treat patients, especially the ones that are in severe cases. We should remember that there is only about 5% of the patients are actually getting to severe complications. So these are the patients that I think we should have the focus on them. Most of the patients will do fine. We are still not aware to the long-term effect of the COVID. But this is, I think, the approach that should be actually applied, and we are trying to change that in Pluristem in what we are doing.
Amazing. Well, I think we're actually getting toward the end of the panel, so I want to thank you very much for being a part of it. Professor Nahmias, Professor Symon, and Yaky, thank you very much for being here. I hope that all of your work goes well, and that next time that we meet around this table, that we have both a vaccine and also the best treatments available so that we can move beyond the pandemic. Thank you so much.
Thank you very much.
Thank you.